All right. So we left off with our slide where we were putting everything together. And where we left off was also, we also left off with not addressing perhaps some of the issues that can emerge from replication, that our body has these endogenous mechanisms to address them. One issue that can emerge during replication is called the end replication problem. So for linear chromosomes like that we have, what ends up happening is that because we have this primer, that if we have this primer on the very end where it helps to trigger DNA polymerase delta to begin elongating, what ends up happening is that once this primer is removed, there's not much space for another polymerase to come in and replace the complementary sequence for this end of the DNA. And so what ends up happening is that with each replicative cycle, we lose that part of the DNA. And so when that's copied, now we have this segment that's shorter. And when there's a subsequent replication cycle for this DNA, there's going to be a primer there. It's going to be elongated. And once that primer is removed, we won't be able to have a new polymerase come in without a primer there, because again, it can only elongate when there's already something there. And so because of that, because there's no sequence there, we end up losing that region. There's an endogenous mechanism that helps to protect and address that end replication problem, where there are these regions of DNA on the very ends that we call telomeres. There are these like repeat base pairs that ensure that we're protecting the actual regions of our chromosomes, the actual DNA sequences that have our actual encoding regions of DNA. And so the very ends, all of our chromosomes, every chromosome at the very end, have regions that are called telomeres. And it's actually really, and what ends up happening is that there are these enzymes called telomerase that will then replace the telomeres and help grow them back out so that they can continue to replicate. Because what ends up happening is that when we lose these telomeres, replication stops. And so the telomeres are actually a great way to basically tell the age of a cell, for lack of a better phrase. Because once you lose that telomere, once you lose telomeres, that begins this like shutting down process. And so the loss of telomeres is kind of a consequence of aging, for lack of a better phrase. And so telomerase, that enzyme, helps to prevent the shortening. And the way telomerase does that is it's actually, this is going to sound familiar, it's an RNA-dependent DNA polymerase. But what's really fascinating about telomerase is that when we talked about DNA Pol 3 or DNA Pol delta, it needed that RNA primer. And so it was provided for it by primase. Telomerase, in the image here to the far right, telomerase already has its primer loaded in. So it provides its own primer. So it's an RNA-dependent DNA polymerase with a built-in RNA template. And typically the telomeres are rich in G's and T's. And so it's like this repeating sequence that protects the DNA away from the telomeres. And so telomerase helps to avoid something called telomere erosion, or the loss of telomeres. And so it ends up providing the built-in RNA template to elongate here. And what ends up happening is it'll enable the protection of the telomeres. And when we lose our telomeres, that leads to something called cell senescence. Senescence is basically, it's not a form of cell death. It's not necrosis or apoptosis, but rather these cells are still alive. They're still metabolically active, but they will not replicate. And so, for example, for those of you who have done cell culture before, for those of you who have worked with either primary cells or non-immortalized cells, you'll have noticed that once you reach a certain point where these cells have grown and doubled and been passaged over time, they basically stop growing. And so these cells become senescent because these telomeres have shortened to such a degree where they can't really replicate any longer. And so one way in which we leverage that in research labs is we can create immortalized cell lines where they have these constantly active telomerases that are constantly rejuvenating or replacing their telomeres, so that these cells are not ever going to reach senescence. These cells will keep going. And so there's some, if you've been in had the chance to work in a lab that's been around for a while, and if you look in the liquid nitrogen tank, you might see cells that have been around since 1987, that have been around longer than I've been alive. And so those are immortalized cell lines that can keep replicating and keep growing because they have this engineered telomerase expression that allows them to continue to replace and maintain the length of the telomeres. Young cells have long telomeres. Yes, go ahead. It's going to be open at the end there, but enough space for a primer to be laid down and recover the telomere, if that makes sense. Because basically, like that RNA primer is a certain length, and so there reaches a point where, like, once that primer is lost, because there's nothing to initiate the new strand there, you're going to lose that region, that primer length in replication. But telomerase will help elongate it, so that when the primase comes in and lays down an RNA primer, now DNA polymerase III has a little more space to replace what would have been lost otherwise. That was a great question. Yes?
So these telomeres are, in essence, also replication?
For lack of, I wouldn't identify them explicitly as an origin of replication, largely because we still don't know enough about eukaryotic origins of replication.
Okay. And then, so do healthy cells substitute telomerase, or is it only for tumor cells?
I would not think of it, the tumor cells is a great question. As opposed to saying healthy cells, think of it as young versus old. Younger cells have more telomerase activity. This is a bit of a reality shock for all of you. We're all adults in this room. So all of us have far less telomerase activity than, I won't say than you did 10 years ago. And so once you reach adulthood, telomerase activity starts to dwindle. And once, well, that's hard to attribute this to specific ages, but a lot of age-related diseases are related to senescence. And that's a very huge open area of research. Like the conference I was at last week had an entire session just on senescence as it relates to scar formation. And so senescence is a very active area of research, but the loss of telomeres is very much age related. Yes.
The long telomerase activity that's in immortal cell lines, is that because of a mutation in the cell line?
Not a mutation, it's engineered. So like we will engineer expression of telomerase to not go away.
About in cell lines where that just existed, like because that was a tumor cell?
Yeah.
That had a mutation that allowed it to.
That's my understanding.
Oh.
I'll go back and double check that, but that's my understanding, that there's a unique mutation with Henrietta Lacks' cells that enabled telomerase activity to continue on, and which is why, unfortunately, her cells were used without her permission, and there are likely multiple labs here that still have HeLa cells in their liquid nitrogen tanks. That answer the question? Last question. Do you have a question? Sorry, I thought you were raising your hand. Great.
So you said that the telomeres are necessary in the part where RNA
we lose that primer, but DNA polymerase III needs nucleotides to help. It needs nucleotides to build nucleotides. Once that primer is gone, it can't do that. So for some reason, and this is still something I have a hard time understanding, DNA polymerase I on the telomeres, it won't do it without, once we lose those telomeres, it's still unable to promote elongation. Well, sorry, forgive me. It's still, when DNA polymerase I does it, forgive me, when DNA polymerase does in like the middle, still nucleotides to the right of it. And so it can use that. It's elongating what's already there. But again, this is going back to a quiz question. DNA polymerase, all DNA polymerases need nucleotides to elongate based off of in the 5' to 3' direction. At the end of our chromosome, this is linear DNA, there's nothing for it to elongate based off of, even for DNA polymerase 1. It does, but again, if it's like if my arm is linear DNA, if the primer is here and it's removed and DNA polymerase 1 is going to act here, there's still upstream DNA that it's elongating based off of. Whereas if it's all the way over here, the primer's gone, there's nothing here. There's nothing for it to elongate based off of. And so we're asking it to initiate, which DNA polymerase cannot do. All right. So we're going to spend some time trying to tackle, we're going to try and solve mustard gas and anthrax for the remainder of the lecture. So we talked about the pathogenesis of anthrax, particularly the deadlier form, inhalation of anthrax. What ends up happening is that the spores are inhaled and macrophages in our lungs will, those are the phagocytes in our innate immune system, they will phagocytose or engulf the spores in the lung. What will end up happening is once that happens, this is a pretty favorable environment for now the spore to germinate and to begin proliferating rapidly. And so once that happens, it's going to go ahead and proliferate to such a degree that it will rupture and destroy that macrophage and eventually reach a point until it will promote cell lysis and its germinated non-spore form and cross the epithelium in our lungs. And this will eventually lead to anthrax in our GI system, in our skin. And so once it crosses the epithelium in our lungs and escapes our macrophages, our immune cells, then it can start spreading rapidly throughout our body. And so that's just because there's, if you think of the lung, I think the lung is a rather elegant organ. There's so many different cell types. There's also lots of capillaries within all the alveoli. So once it crosses the epithelium, there's a capillary right there that it can get into and then get into the bloodstream and spread rapidly. And so that's particularly why inhalation of anthrax can be so deadly so quickly.
So in the macrophage doing its job, it's literally giving it the perfect chance to germinate.
Exactly. Yeah. And so that's another reason why once there's awareness of exposure to anthrax, regardless of the symptoms have presented yet, it's important to take antibiotics right away, because with inhalation of anthrax, once symptoms start to show, mortality rates are very high in a matter of days right after that. Yes.
So for bacterium to replicate, it has to remove itself from the spore form.
Yes.
Why isn't MHC class 1 receptors or proteins picking that up and display that?
So what ends up happening is that, again, MHC class 1 is sampling contents within the ER. And so like that's when MHC complex is being loaded, and it'll take, and the ER is like the ER is part of our protein synthesis translation process. And so basically taking things that we're making and sampling it and putting it up. And viruses will hijack our DNA and protein synthesis machinery to replicate themselves. And so for I keep going back to this example, for SARS-CoV-2, we're helping it make more and more of the viral machinery, including like spike protein. For a bacterial spore that germinates intracellularly, it's not in our ER, it's in the cytoplasm here. And so MHC class 1 isn't going to be able to sample that bacterium. And so that's why it's so important to take antibiotics. And we're going to talk about how the antibiotics work that targets anthrax. But that's why it's so important to take the antibiotic as opposed to relying on our innate immune cells to try and sample it and display it, if that makes sense.
Gotcha. So is that the same reason why cells are not involved? Or that's
Different mechanism. Yeah. So that's a good segue. So what's key to address anthrax infection, particularly inhalation of anthrax, is taking antibiotics. And we're going to take a moment to define a few terms that are key for these next few slides as it relates to antibiotics. So there's a whole class of chemicals that are both either naturally derived or synthetically derived, called antimicrobials, that are meant to try and eliminate or eradicate some sort of pathogen. And so that could be some sort of fungal, bacterial, or viral. Antibiotics in particular are synthesized by microorganisms. So these are naturally derived, and they're against other microorganisms. And so antibiotics are under the umbrella of antimicrobials here. And typically we derive for the synthetic substances with microbials. So typically antibiotics are usually naturally derived. For antimicrobials, some of them are synthetically derived. And those that are synthetically derived, we usually produce them, or they're usually discovered either through some sort of screening process or also through chemical synthesis. So there are some pretty desirable properties we have for antimicrobials. And so these next few slides, in general, I know like about a third of the class is pre-med, and so this will probably help or pique the interest of the pre-med students in the class. So there are some specific desirable properties for antimicrobials that we usually look for. We want there to be selective toxicity, that we want it to attack and address the invading pathogen or the microbe and not the host. And so typically, antimicrobials will target something that's unique or specific to a pathogen that's not present and are present within our own biology. They're usually also pretty, we want them to usually be stable. When we talk about stability, we talk about being able to not degrade at temperatures when we store them at. And so room temperature is great, not always feasible, but having good shelf storage, having stability on shelf storage is key. Being cheap and easy to administer. This next part is key. We want it to be bactericidal and not bacteriostatic. What that means is we want it to kill every bacterial microbe and not for it to be bacteriostatic, where their growth becomes static or we stop their growth. So we don't want to just stop their growth. We want to kill every microbe within that microenvironment. So if we had a tube of, say, like 1,000 spores of anthrax in a tube, and if we had a drug that we delivered, or 2 tubes, I should say, and we had 2 drugs, one drug at a certain time point, say 7 days later, if there was 0 spores present, that would be bactericidal. It's killed all of them. After 7 days, these spores would, in a favorable environment, grow to thousands to millions of spores. If it stayed at 1,000 in this other tube, based on this other drug would be bacteriostatic. It stopped their growth. We don't want that because that'll enable for antibiotic resistance. And so we want bactericidal drugs. That's the desirable quality. And that addresses the next point. It should not become easily resistant to the drug you're developing. And so if it's bactericidal, it won't be resistant. We want there to be appropriate distribution. If you have a bug in your GI, taking the antimicrobial orally would make sense. If you have a UTI, an oral delivery would not be ideal, probably. We want there to be no adverse reactions like allergies, which is a huge concern for antimicrobials. And we want there to be an appropriate spectrum of activity. And that spectrum of activity typically refers to what it acts upon. And we're going to talk a little bit more about that now. And so you've probably heard the phrase of broad spectrum versus narrow spectrum antibiotics. Broad spectrum antibiotics typically will indiscriminately try and kill every single bacterium within its path, which is not ideal because we also have our commensal bacteria within our GI tract, as well as in other mucosal layers that we need. We have, sorry to say this, pounds of bacteria within our body that our body relies on. That probably sounds a little gross, but it is the reality. And so it's much better to try and have narrow spectrum antimicrobials that'll have a more targeted approach. Sometimes it can be, for example, we already talked about gram-negative versus gram-positive bacteria. And so that's one way in which you can have a bit more of a targeted approach. And another, we're going to talk a little bit more about different ways you can have a more targeted approach based on what you target. One consequence as a result of broad spectrum antibiotics is something called superinfection, where if some sort of new infection, if there's some sort of existing infection that occurs, and you have a huge broad spectrum antibiotic that lays to waste every bacterium in its path, also the good ones, what's going to end up happening is that new subsequent infections will end up developing resistance to the drug. There's going to end up being a selection for those that can survive that broad spectrum antibiotic, and will eventually lead to subsequent really problematic infections. One example of that is C. difficile. That is usually one that crops up in hospital settings when broad spectrum antibiotics are used, and it's this gut infection that can be really dangerous. One example of one analogy, I want to come up with a better analogy than this, but this was created for me. If you have a specific ecosystem and you kill all the predators, what ends up happening, there are consequences for that in this ecosystem, where if all you have left are deer, sheep, and cow, for lack of a better phrase, within this ecosystem, this very narrow ecosystem, you'll end up having animals that will eat all the grass, all the shrubbage, and it's going to destroy one element of the ecosystem that's going to create a total imbalance and eventually lead to destruction of the ecosystem. And so what the whole take-home point of that is that broad spectrum antibiotics is superinfection. When you destroy all of the microbes within your body using a broad spectrum antibiotic, you're destroying all of the bugs, including the good ones, and creating an environment where only those that are resistant will continue on, which will have subsequent consequences that are even more troubling. So antimicrobials, as they're designed currently, some of the ways in which we've created narrow spectrum antibiotics is by targeting specific qualities of bacteria or of microbes that are unique to them, that are not present within us. And so, for example, we do not have a cell wall. So we can develop antimicrobials that are cell wall inhibitors. One example of that most people probably have heard of is penicillin. Another one is a cell membrane inhibitor. And so fungi have a membrane and qualities of their membrane that's unique to them, that's not present in our cell membranes. And so for antifungals, there's cell membrane inhibitors that enable us to target them. And typically there are these things we're targeting that are very essential. And so that's whether it's a cell wall or cell membrane or some of these other processes we're going to talk about. But some antifungals target cell membrane inhibitor or are cell membrane inhibitor for fungi. Some that we're just going to gloss over quickly are protein synthesis inhibitors. We haven't talked about translation yet, so we're not going to dwell on this too much, but some are able to actually try and affect the protein synthesis pathway in microbes. Some of you who work in labs might have heard of streptomycin before. We use that in our cell culture media. There are also metabolic enzyme inhibitors that try and target specific qualities that are unique to metabolic pathways and pathogens. The ones that we're going to talk about today are ones that inhibit replication, but there are also ones that can also target transcription directly or indirectly as well. So the one we're going to focus on for today is ciprofloxacin, or just called Cipro. It is considered one of the most powerful antimicrobials around, and it remains to be the only effective treatment for inhalation of anthrax. Excuse me. It's within a class called fluoroquinolones. That's a class of antimicrobes that are determined based on their functional groups and their chemical structure. And so you usually won't see anything like this naturally derived within us. but what it does is it specifically inhibits bacterial DNA gyrase. Bacterial gyrase is different than our gyrase, and so it allows us to target their gyrase. And if we can inhibit, if we inhibit their gyrase, we're disrupting the replication, and if there's no proper replication, there's no division, no growth, and we can destroy bacterium that way. And I just scooped myself for the next question of, based on what we know about replication, what will happen next. But what this looks like is if we have ciprofloxacin that is disrupting their gyrase, what will end up happening is as replication will emerge, not able to address, and so the replication work will basically no longer be able to extend. The polymerase won't go any further because it's hitting an actual physical barrier with this supercoiling that's not being alleviated. Before I'm going to stop for a second, wait for a couple of questions here. So as I understand it, this is bacteria. I'm going to look into that and not venture a guess. My hunch is that it's bactericidal because of how dangerous anthrax is, but I will double check and confirm whether Cipro is bactericidal, because I want to say I'm fairly sure it is, but I don't know off the top of my head. Oh, and before I forget, there was a question last class about DNA, why do prokaryotes have a gyrase that sometimes melts DNA? Does that sound familiar? So basically because there's one origin of replication for prokaryotes, whereas we have many. And so if it's inefficient at melting DNA, when there's just like one spot where it's going to be doing that, that's okay. But because we have so many origins of replication, we can't have an inefficient melting of our DNA. And so we need like a professional helicase to do that. Does that help answer the question? I hope I'm pointing to the right person right now. Okay. I've made that mistake before. Yeah, any other questions? Go ahead.
So, so when we say it's a very powerful antimicrobial, why do we use this all the time? Is it just because it's very broad acting?
So again, we want a narrow spectrum that addresses the specific bacterium that's at play here. And so if there's you can think of it as like defining the path of least resistance for how we can tackle certain bacteria based on like where they are, based on, so for example, like if you have a GI infection, or if you have a upper airway infection, there's different delivery routes, and the stability for some of these antimicrobials, like how can they survive some of these delivery routes? Like if some of them can't survive or have proper delivery with like an oral delivery, that becomes a problem. So like some will and some won't. And so it becomes almost like this process of determining which ones can have proper delivery to the site of infection. And based on the species of bacterium, and usually like, they'll do a culture to figure out what you have. And based on what you have, these are the proper antibiotics that we'd suggest as a result of that. And so it's all, for lack of a better phrase, the simplest answer is that it's all context dependent. If that makes sense. Because again, you don't want to have a just like broad, you don't want to deliver a fairly broad antibiotic here because again, that's how you're going to potentially lead towards resistance here.
So what makes Cipro so specific to anthrax? I would assume most bacteria have Toplas operators.
Correct. But again, this is a fairly strong antibiotic. And so you wouldn't want, I'm trying to create, I was going to try and use a violent analogy, but I'm not going to go that way. You typically want to try and make sure that you're not going with the most aggressive path first. For lack of a better phrase, you want to work your way up towards that. If there's a very narrow antibiotic that you can go with first, you go with that path first, as opposed to, for lack of a better phrase, going in with the big guns and going in with a really broad, not like you said, all bacteria have DNA gyrase, and so you don't want to have this like really broad spectrum antibiotic that's going to be problematic. And so because anthrax is so deadly, it's either death or a super powerful antibiotic. And so that becomes the decision you're weighing, if that makes sense.
Gotcha. So it is broad acting.
That's a fair way to put it. All right, so now we're going to solve mustard gas. So we described before how mustard gas is a vesicant. It's a blistering agent. We don't quite know how it's a blistering agent, but what we do know is that the keratinocytes within our skin are pretty vulnerable to this effect. And so it creates this blistering effect. But what's really interesting is that there's a DNA effect as well. And what that DNA effect is that particularly it affects, modifies guanine. What that looks like is that it'll alkylate and cross-link adjacent guanines. So it will lose the chlorine groups on each alkyl chain and attach to the nitrogen as a nucleophile and cross-link them together there. And there's a few ways in which this will have pretty severe repercussions within our DNA. And so the effect of mustard gas on replication, where it cross-links guanines together, we can have intra-strand cross-links and two adjacent guanine cross-linked together. We can have a standalone guanine. There's an alkylation where there's the alkyl chain hanging out. Or more dangerously, there is interstrand across strands of cross-links of guanines. And so what ends up happening is when you have your DNA polymerase reaching an intrastrand crosslink, this will create a physical barrier that halts and stops the polymerase there. Same thing with this alkyl group here, alkylation event, where it'll, again, it's another actual like physical disruption based on the chemical changes here. But what's really quite potent is when you have these interstrand crosslinks, when helicase is trying to melt and separate the DNA, when there's an interstrand crosslink, it is unable to. And what ends up happening is that this will, you'll have a replication fork collapse as a result of that, because it's not able to, the helicase isn't able to go in and, for lack of a better phrase, open up or melt the DNA. And it's going to lead to something called a double-stranded break in our DNA, which is really dangerous. And so because these double-stranded breaks are quite problematic, some have considered using mustard gas as a chemotherapy to fight cancer. Because it will be able to target and halt replication in cancer cells, it was, again, considered to be a chemotherapeutic agent. But I'll pose a question to the class of why do you think mustard gas, which affects replication, would have been a good idea as a chemotherapeutic? It's not a trick question, but like, why would that be a good idea? Yeah. Yeah, exactly right. There's unfettered growth in tumor cells. And so using something that targets replication would be particularly helpful. As you can imagine, there's not really a targeted way to use mustard gas so that it only affects tumor cells and not your healthy cells. So it becomes a bit of a problem. But in addition to effects on our DNA, there's also naturally occurring base damage. In response to UV rays, in response to radiation we get from different sources, in response to different injuries, we get about 25,000 lesions per, per year, per hour in our genome. And so some of those alterations in our DNA are, there is a depurination event where basically we lose on guanine, we lose the purine group here, and we just have a sugar, a depurinated sugar here that would be considered a base damage to our DNA. There'll also be a deamination event here on cytosine where we lose this amine group and it goes from being a cytosine to uracil. It goes from being DNA to RNA. And other forms of naturally occurring base damage have, in response to ultraviolet light, adjacent thymines will be crossing together and we'll get a thymine dimer. And so all of these are different ways in which base damage can occur. And if we're getting so many potential base damages per hour based on everything that's around us, based on whatever degree of radiation we have, or if any of you become astronauts and go into space, even more cosmic radiation, how do we have repair mechanisms to fix that? And we do. We have what's called base excision repair. We talked about the exonuclease activity of DNA polymerase, where it can proofread back up at the ends of this growing strand. So that was an exonuclease. For base excision repair, we have these endonucleases. Remember, they work within the middle of a DNA strand. So these are repair endonucleases, where they're able to find an error, this physical disruption within our double-stranded DNA, likely, again, like a physical disruption, and remove this damaged region with the help of helicase if needed to. And what ends up happening is, because after that's been removed, damaged DNA is removed, because there is an upstream region of DNA that that DNA polymerase can elongate from, it'll go in and fill that excised region to be complementary to the anti-parallel strand, and then the ligase will fix and repair the separated backbone. And so these repair endonucleases are really key for making sure that whenever some sort of lesion or change occurs to our DNA, which is happening all the time, these repair endonucleases are going in and fixing those problems. Now, as I mentioned before, the double-stranded break in response to mustard gas, also in response to radiation, also in response to cosmic radiation, again, if any of you are lucky enough to be astronauts, will cause not a lightning strike, but radiation that will then actually be enough energy to create a double-stranded break, which again can be quite problematic if not taken care of. We actually have two different endogenous mechanisms that are double-stranded break repair pathways. We're going to talk about both of them and talk really briefly about how this is also leveraged in genetic engineering. And so when there's a double-stranded break, one pathway that tries to fix that is non-homologous end joining. What ends up happening is that we have these two free ends at this double-stranded break, and there are proteins that recognize this double-stranded break called end-recognizing proteins. And there are these slight overhangs across these strands. And what ends up happening is that these end-recognizing proteins will bind and help try and sample the overhangs without using a real template, to just try and provide a few nucleotides to just fill the gaps and to try and create fill the gaps and rebind using ligase these broken ends. And so the first key thing here is that there's not a key template being used here to try and fix and repair, but rather these end recognizing proteins will try and help sample and bring in nucleotides to try and fill in the gaps And sometimes there can be small changes. Like this might sound quite similar to VDJ recombination, where there's like this sampling and there's like slight changes that occur. With non-homologous end joining, it's not too dissimilar, actually. The other pathway is called homologous recombination. This is actually a really elegant way of trying to fix these double-stranded breaks. And so when there are these double-stranded breaks, and again, there are going to be these overhangs, it actually is a process that leverages that we have 2 copies of every gene. We have two chromosomes. And so what it'll actually do is use the other chromosome as the template to repair these double-stranded breaks. And what ends up happening is that one of these segments will go in, and there'll be a homology search across the other chromosome to find the complementary strand. And once that does, it'll go in and helicase will go in and open in during this homology search. And once it finds it, it'll use the other chromosome as a template, repair via DNA polymerase and elongate that chromosome. And once that happens, then DNA polymerase can go in and fix this end on the other strands of that double-stranded break, and ligase repairing them. And so the key distinction between non-homologous end joining and homologous recombination is that non-homologous end joining does not use a template. It doesn't use the other chromosome as a template here. It's just using these end replication proteins to try and help use a few nucleotides here and there to fix this double-stranded break. Homologous recombination is actually using the other chromosome as a template to try and repair and fix. And usually both copies of chromosomes should have same copies, not always, but same copies of the DNA. And so it'll use that as a template to fix, repair, and re-anneal back these double-stranded breaks. What's really fascinating is that this whole mechanism is leveraged with the genetic engineering tool CRISPR-Cas9. So to try and take a moment to describe what's happening here is that there are these, we have both Cas9, which is an endonuclease. So again, it goes in and will make repairs at specific regions within the DNA. And what's really fascinating is that this allows us to have a really targeted approach where we have, it's an RNA-dependent, so it has a template provided. Usually in a research setting, we provide that RNA template. We call it a single-stranded guided RNA. And what's really fascinating is that the black region is what we call the CRISPR RNA. To give some context, all of this was discovered not too long ago. It usually takes decades for something to be recognized by the Nobel Committee for a Nobel Prize. A lot of this was discovered by a professor at Berkeley named Jennifer Doudna in 2013 with the help of another researcher, and another professor, forgive me. And in 2017, 4 years later, they won the Nobel Prize for this, because this is a really powerful genetic engineering tool that was really cost effective and really efficient. And so what ends up happening here is that the Cas9 endonuclease was first discovered in archaea, these like deep sea organisms, and CRISPR, I had the acronym in my head and I've lost it. It's basically these clustered repeats within DNA that were present within bacteria whenever they were infected by some sort of virus. And what they would do is they would take these repeats of that virus's DNA. This was their adaptive immune system, basically, and they would insert it into their DNA. They would edit their own DNA, insert what was present from that virus, so that when they have a subsequent infection, they'll remember that virus. And so these researchers discovered these two mechanisms, and were able to see that if we combine them together, and if our cells express Cas9, when we deliver a CRISPR RNA that will then be loaded into this Cas9 endonuclease, what will end up happening is that there's going to be this protospacer adjacent motif, the PAM motif here. That's kind of like the initiation site. And the protospacer allows us to have a very targeted approach for where this is happening. And so this was typically happening in bacteria in a very broad sense. But when we provide this sgRNA or the single guide RNA, we're able to target it specifically to the protospacer here. And what ends up happening is that there's going to be a double-stranded break because there's two there's going to be two molecules that help mediate the double-stranded break there. And two things can happen here, either the non-homologous end joining or the homologous recombination. With the non-homologous end joining, it's actually a way for us to try and be able to leverage some of the mechanisms where we can delete a gene, and there's going to be a few nucleotides inserted here and there. But with the homologous recombination, what ends up happening is that not only are we able to delete genes now, we're able to cut and replace them with what we want in there in a really cost-effective and efficient way, where based on single guide RNA as a template now, combination will use that as a template to write in a copy of the gene as we see fit of how we want it to be expressed. And so this is a really powerful tool that, again, it's effective, it's cost-effective, it's efficient, and it allows us to rewrite genes in specific ways. And so when we talked about beta chain of hemoglobin in the very first lecture, this is now being used in clinical trials to try and replace hemoglobin expression so that it doesn't promote that sickled allele. This is not yet in clinical trials for cystic fibrosis, but hopefully a couple of projects in this class will help spur that on. And with that, I've run a couple minutes over, so I'll go ahead and stop it here. But if you have any questions, you can come up and join me. If you have any interest in DNA replication, here are a few folks that are present in our university community.